So we're going to talk to you guys today about some of the work that I'm doing in Professor Judith Hoyland's lab as a Athletics Research UK Foundation Fellow. So the work that I'm doing on is, the aim of it is to try and hopefully start to develop a stem cell therapy for repairing intervertebral disc disease or degenerate intervertebral discs, which is one of the main causes of low back pain. Right, so this slide is just to go through some of the stats surrounding low back pain. It's kind of like classic clinical slide to try and convince you that it's a really important disease and it's worthwhile actually putting lots of money into the research to try and come up with a therapy to stop this. So it's believed that it affects around 85% of the population at some point, so at some point during the duration of their lives. It's estimated to be the second most common reason to visit a GP, with the first being skin-related complaints. 10% of sufferers are chronically disabled and accounts for around 50% of all sick leave. A certain percentage of that will be people pretending to have low back pain as well. It's quite hard to work out which of the people who present to GPs with low back pain are actually just lying about it and which ones are not. So in terms of financial costs, it's believed to cost around 12 billion UK pounds. This is a year, or around $200 billion in the US per year. So again, it's hard to put an exact figure on the cost because although a lot of this comes from lost days in work, some of it will be people just skiving. But it's still a large proportion of cash. So what are the causes of low back pain? So they are definitely multifactorial. However, in the majority of cases, the intervertebral disc is involved. So this can range from acute injury occurring in the disc, so this could be from some kind of trauma, car crashes, things like that, where you have acute damage happening. Or it can be in terms of degeneration, so it's a bit like osteoarthritis happening inside the intervertebral disc. I'll go through more detail about it in a minute. So in terms of the disc, it includes disc herniation, so this is where you would have a bit of disc tissue actually pop out, and the disc then stimulates the spinal cord, and so you get pain resonating down from that down the legs as well as up. Myelopathies as well, so that's some kind of inflammation occurring to the spinal cord because of disc herniation. And then also spinal stenosis, which is a narrowing of the spinal canal and just starts to close off the spinal cord. Intervertebral disc degeneration is quite strongly associated with low back pain. Something around 40% they think of low back pain, or real low back pain, is associated with disease of the disc. So, just want to kind of go through what the healthy intervertebral disc should be like. So this is a picture of the spinal, of a spine. So humans have 23 intervertebral discs located all the way down the spine. So the discs separate the vertebral bodies, but they also act as shock absorbers between the spine. So instead of the body could have evolved some kind of solid, maybe or kind of in socket type system, but it's gone with this system we believe is, for a certain extent, is to do with the shock absorbing capabilities of it, as well as providing motion through all the different planes. So inside a healthy intervertebral disc, you've got two main parts to it. So you've got the central gelatinous region, which is this bit here, which is termed the nucleus pulposus. And then to the periphery of that, you've got the annulus fibrosus, which is made up of like cartilage rings. It's a bit like tendons really going on the outside. So when the intervertebral disc is loaded, you've got pressure coming above and below, and you have the central gelatinous part of the disc trying to expand outwards, and it's the job of the annulus fibrosus to constrain the expanding nucleus pulposus, and that's where it gets its kind of force from, which is a bit like a pressure pad. So the nucleus pulposus is quite similar to articular cartilage in terms of the fact that it has no vascular growth within it, or any nerves either. So in the healthy disc, there should be no blood vessels or nerves within it. The matrix is made up of proteoglycan and collagen, So these are important in providing all the mechanical stability and mechanical function to the disc. And it's the proteoglycans, which are negatively charged, which are able to imbibe water, and so you get very hydrated matrix, and that's where a lot of the mechanical function comes from. It's hypoxic, so low oxygen, low pH, and also low nutrients. So it's quite different from a lot of other tissues in the body, which do have a direct blood supply. Not a lot of cells in the tissue either compared to others, and the cells within it are quite similar to chondrocytes, so similar to cells found in articular cartilage. And we know this because they express a lot of the same markers, SOX9 transcription factor, as well as the important matrix proteins. So in this disease, we know now that it's actually driven by cell processes. So in the normal disc, you've got this nice hydrated tissue. And what's happening is that the normal intervertebral disc cells are maintaining the tissue. So there's a nice balance between synthesis of matrix as well as degradation and removal of older matrix or damaged matrix. But with the onset of degeneration, you get an imbalance between the synthesis and degradation rates. So it seems to be a shift towards matrix catabolism. You get a decrease in the synthesis of healthy tissue and an increase in the enzymes which used to be used for just remodeling. So as the disease progresses, you get kind of change from a nice hydrated tissue and matrix to this much less hydrated. You can see it's actually been, blood vessels are starting to grow in. It's actually red in colour there. So you get matrix degradation, which begins in the nucleus pulposus and then kind of works outwards, and you get these cracks appearing from the annulus fibrosus. As the matrix gets lost, the ability to imbibe water decreases, and so the tissue starts to dehydrate, and you get a loss of disc height as well. With the loss of disc height, you get a loss in the mechanical properties of the disc. So current treatments are mainly considered to not be adequate. So in terms of medication, these are in the form of painkillers, and so they're addressing the pain associated, but not actually any of the function or loss of function that's attributed to the disease. So while these can start off, they can be useful at the beginning because they will halt the disease. As it becomes more and more progressive, the actual amounts of pain can increase until the fact that the pain relief doesn't work anymore. In terms of non-invasive therapies, things like bed rest, spinal manipulation, physiotherapy, these have been proven to help in this situation. However, if the degeneration and the pain associated and loss of function is too great, then you have to go on to more invasive therapies. So these involve things like discectomy, which is just removal of the diseased disc, and then you get fusion. So this is where they'll put metal bars into the vertebral bodies above and below the diseased disc, and that will just basically remove the motion segment, so you start to lose any kind of flexibility in the spine. And although this does provide good pain relief for at least a couple of years, you get this destabilization happening in the spine, and therefore the degeneration appears to move above or below what was the problem disc. So there's also disc replacement therapies as well. So this is where you actually have a motion segment popped in. But the issues with this tend to be just wear and tear happening, so you'll have an operation within a few years, either the piece of metal or rubber, depending on what it's made of, will start to move, and it's meant to be kind of fixed to the vertebral bone of bodies. It can actually come away from that and become an issue. And if you've got something like this starting to move around, it's very near to the spinal cord, so you can imagine that it's quite high risk if that happens. And then you also get, with wear and tear, very small particles coming off, which can cause inflammatory response as well. So currently there is a lack of successful treatments targeted towards the actual disease process, which is why the lab is interested in trying to develop a stem cell therapy for this. So as I mentioned before, the degeneration and then the associated loss of disc height are cell-driven processes. So how this happens is in the normal tissue, you've got an MP cell, and that will be maintaining a nice hydrated matrix. So something then appears to happen with disease. We're not entirely sure what this is yet, but it appears to be a change in the environment. So this could be increase in inflammatory cytokines, or perhaps a decrease in pH, or a drop in nutrients. So the disc is already limited in terms of the amount of nutrients in it and has a low pH. But this, we think, can get progressively worse during disease. As this happens, you then get synthesis of cytokines. So these can be things like TNF-alpha, which drives catabolic processes, as well as neurotrophic factors, and also things like VEGF, angiogenic factors. So as these start to increase, you then start to get an increase in the vascular growth into the disease disc. And as blood vessels grow into the disc, it's been shown that neural cells and nerves actually grow into the disc as well. That can increase the pain. We also have increase in other cytokines, and this leads to cytokine-mediated events. So these include things like altered matrix synthesis. So this is where this imbalance happens between maintaining a nice balance, homeostasis, and actually shifting towards a catabolic response. So we have an increase in some of the enzymes, which are usually just used for maintaining the tissue. These enzymes go up, the synthesis of the matrix proteins goes down, so we get a shift. We also have cell senescence happen, and that causes an abnormal cell phenotype, as well as apoptosis, and then as the disease progresses more, we actually have lots of cells within it. So the strategies that we think could be used for trying to repair some of this degeneration in lower back pain. We've got three main ones. So the first one, we're trying to target early degeneration. So this is where there are changes in the cell phenotype, but there's not any cell death, so there's no need to actually inject in any additional cells. We can just put in biological inhibitors into the disc by injection, and these might be able to try and shift the imbalance towards catabolism, try and shift it back towards anabolism. So for instance, one molecule that the group are interested in is interleukin-1 receptor antagonist, which can inhibit interleukin-1 and actually start to reverse some of the catabolic associations with that. If the degeneration is worse than what we could use for at the early stage, then it may be that we actually have to start putting in cells into the disc. So what the group would be thinking about there is to try and take some kind of cell, maybe include it in a biomaterial, and then inject that into the disc, and hopefully the cells that you put back into the disc can take over the job that the old cells used to be able to do. And if the disease is really far progressed, then maybe it's gone beyond that. If this isn't intact anymore, then we might actually have to try and engineer a disc externally and try and use that as a way to sort out the low back pain. So some of the important questions that the group need to answer in order to develop a cell therapy are what is the most suitable cell source? So we can't just take nucleus pulposus cells from a patient suffering from low back pain. Because even if we go to a disc where the disease hasn't begun, if you go in and try and remove cells from that, you're going to initiate degenerative processes. So the group are interested in adult mesenchymal stem cells because they're easily accessible. People have shown, including our group, that we can differentiate them towards the cell type that we might want. Other members of the group are also trying to look into what a suitable biomaterial might be, so looking at different types of hydrogels, and whether we can use a hydrogel to help the differentiation of the stem cells towards the cell type that we want without having to use growth factors, which should keep the cost down. And also, are we definitely able to get mesenchymal stem cells to differentiate to MP cells, and how do we know that they are becoming the cell type that we want them to be? And then lastly, is the environment of the degenerate intervertebral disc, is that going to be important? So when we put the cells into the disease disc, is that going to have some sort of influence on the cells? So that's the bit that most of my work is focused on. So I'm just going to introduce what mesenchymal stem cells are. I'm not sure whether you guys will be familiar with it. Most people have heard of embryonic stem cells, and they are able to turn into pretty much any cell within the body. So the mesenchymal stem cells are multipotent but not pluripotent, so they can't become any cell in the body, but they can become mesenchymal cells. So these include things like bone, cartilage, muscle, and also intervertebral disc cells. So these are useful. One of the reasons they're so useful is that you can acquire them easily from adult tissue. So you haven't got any other controversies surrounding them like with embryonic stem cells. So I just want to talk briefly about the effect of the intervertebral disc niche on MSCs. So inside of the diseased disc, as a degeneration occurs, the environment, which is already hostile in a normal disc, starts to become progressively worse. So you've got things like the nutrients is very low, it gets lower, low oxygen, low pH, high osmolarity, high levels of cytokines, and then also the fact that these tissues are getting loaded a lot as well. So with progression of disease, this harsh environment becomes even more harsh. And the work that I'm focusing on is the effect of low pH, because there's some evidence to suggest that this may be the most detrimental aspect of the disease disc. It might be driving the disease process. So this is just some data. I'm not going to go through a lot, but just to show that when we do treat mesenchymal stem cells with progressively lower pH, this is going down to the pH that you might find inside a diseased disc, as well as around kind of this sort of level. And 7.1 is in a healthy disc. You find that you do get a decrease in the proliferation. So the cells appear to not proliferate at all at 6.8, and then actually start to die off at pHs below that. And by the time you get down to 6.2, quite a lot of the cells are dead, but not all of them. So we're quite interested to try and work out what is it about these cells which allows them to survive at such a low pH while the other ones are not able to. When looking at proliferation, it's just looking at the DNA content. So we found that after 1 day of culture, that the dark line there is the seeding cell number. That you find as the pH drops down towards 6.2, you do get cell death just after 1 day, and that's more apparent after 7 days. So we've also been looking at whether, because some of these cells are still alive, are we able to select for those cells or try and grow them after that? In terms of looking at the effect that pH might have on the gene expression of stem cells, this is just some data looking at a pain-related factor. And you can see that when you treat the stem cells with progressively lower pH, just after 1 day, you get an increase in pain-related factors. Whereas if you treat them for up to 7 days, that drops away. So again, another pain-related factor, you get an increase at day 1, but then that drops away at day 7. With this one, you have an increase, but then it drops. Actually, it starts to drop away completely by day 7 and goes much lower than what did originate at pH 7.4. So we think that this is quite interesting because it suggests that if you're going to implant mesenchymal stem cells into a diseased disc, if you pop them in, within 1 day, they're going to start kicking out a lot of these pain-related factors. And that could have detrimental effects inside the disc. Whereas if you pre-treat the cells in low pH for maybe 1 week and then implant them, the cells are much less likely to start releasing pain factors and maybe actually behave much less in an inflammatory way. So this has led on to some more important questions that my work is going to focus on in the future. That is, why do some of these stem cells survive in the low pH and how come other ones don't? Do we need to be preconditioning the MSCs in low pH prior to implantation in terms of can we treat them in low pH for a week and then are they going to be much more useful for actually regenerating the disc? And can we select out for the pH-resistant MSCs? So do we have to do that by culturing them in low pH, seeing which ones survive and then growing them up? Or can we find what's special about the cells which survive and then select out and maybe using some kind of surface markers? So So in summary, intervertebral disc degeneration is a cell-driven process. The MSCs inside the MSC's function is influenced by the intervertebral disc niche, so it is important to try and to consider that in order to progress the stem cell therapy towards clinic. And that's been evidenced in terms of in vitro pH studies. So future work will look at preconditioning the MSCs against a low pH to see whether we can try and have a more suitable cell source. And all of this is aimed at trying to translate clinical therapy towards regenerating the disease intervertebral disc and trying to treat low back pain. If anyone's got any questions, I'm happy to answer.
Any questions?
Did you say that low pH causes the degeneration in the first place?
Yeah, there's no direct evidence for that, but recently more and more evidence does suggest that, and so I do believe that that's probably what's happening.
Okay, so if you implanted the MSCs with
Yeah.
would it not degenerate the pH when you change the pH?
I think there's two. Either you have to change the pH, or you have to make sure that the cells are different from the cells that originally were in the disc. So I think changing the pH is going to be too difficult. So what I would hope to do is that by preconditioning these MSCs in terms of finding which ones can survive in the low pH, looking at their phenotype versus the cells which died off, that we can select out a cell type which will survive in low pH.
So it's like changing
Yeah, so we'll know what type of phenotype we want them to have in terms of what structural proteins we need them to be making, and what enzymes we need them to be making to have natural turnover. But we need to try and find a way to have them have that correct phenotype when the pH starts to drop away. Yeah, because during kind of the process of disease, it seems to be that the nutrients transport into and out of the disc starts to get progressively worse, and so you do get this drop in pH. So the only other way around it would be to try and prevent that nutrient transport from getting worse. I'm not sure how you could do that, but another option there.
So in terms of trying to condition cells that are explaining at a low pH, although they might survive, do you think it might be a problem with formation of proteins? So, as you said, you want them to produce the right structural proteins in the IVD, so it regenerates the tissue, essentially.
Yeah.
But do you think that maybe the effects of the low pH on protein formation would mean that they won't form proteins even though they can survive?
Yeah, it's definitely that could be an issue. It's whether the pH of around 6.5, 6.2, whether that is too low for normal, after the proteins have been synthesized, for them to start to conform correctly. I'm not sure where the cut-off point is going to be there, but it probably would have an effect. So again, that would be another obstacle to get around as well.
This is probably going to be a few questions. When you say that the small population that survive at low pH, and then what you want to look at to see how they survive, how are you going to sort of target them? Are you going to be using the single cells?
So, one thing I've been doing is that when you treat with the low pH and you get the majority of the cells dying off, I've had a go at just switching the media back to a nice normal pH. You find that those cells then proliferate. So it's whether I can just use that as a population of cells, at least to begin with, whether I need to be looking at individual cells, maybe like clonal studies, something like that, to find out which ones are surviving, which ones are dying, what is different from that. But it suggests, as does a lot of the literature, that it's well known that mesenchymal stem cells exist in like a heterogeneous population, and they're all probably working together. And if you select out just a particular cell type, as it were, within a particular group of MSCs that are expressing particular cell markers, you'll find that they will differentiate back to being heterogeneous. So they like to be a mix because I guess they're all doing different functions.
Thanks for the talk. I was particularly drawn by your statement that is it the NP cells? You say during degeneration something happens in the cell which probably scientists don't have an understanding of. Now, that being the background, is there any parallel work looking into what has happened to that? Because if you don't know what has happened to that cell, you might be bringing cells to an environment. Maybe it's because by the environment you bring whatever cell comes in there, it's not going to work. Is it not therefore the way forward to look into what has happened? Because for us to establish a cause-effect relationship, we need to have our grip on what is.
Yeah, so within the group, we also have a certain number of scientists working on like what the etiology of the disease is and how that is having an effect. And when I say we don't understand, that's probably more because I've worked in the field for a few years, and so I feel that really we still don't understand.
Many processes, we don't understand
idea,
many processes, yeah. I mean, it's true.
Yeah,
We understand very few, yeah.
We know things like there's elevation in some of the inflammatory cytokines, and that seems to be driving a lot of the degenerative and destructive processes that are happening in the disease. But we know that there's, for instance, high interleukin-1, but does that come after something else, or is the high interleukin-1 driving it? Not really sure. I found that when I put MSCs or nucleus pulposa cells into low pH, they increase the expression of interleukin-1, which is why I think it may be the low pH which kicks off the increased amounts of inflammatory cytokines, and that has a knock-on effect in terms of shifting towards a more catabolic phenotype. But again, that's not been published, and it's my thinking. And yeah, there's lots of other contributing factors, I think, as well. But yeah, you're right, it's important to understand the disease process, and then hopefully at the same time trying to understand whether we can develop a therapy as well.
So, I mean, this is probably a stupid question, but is the pH contained within the disc? Like, is there how much it can react to anything, or is it?
I guess up until the disease gets too progressed, the disc is still a sealed unit, as it were. So you've got nutrients and waste products flowing into and out of the disc from above and below. So there's a cartilage end plate, which is a bit like articular cartilage, and you've got blood vessels which run into the vertebral bodies. So the nutrients flows between the blood vessels from the vertebral bodies down and up into the disc, and then the waste products from the anaerobic respiration, which is occurring because there's such low amounts of oxygen, that diffuses back out again. So you have, even in a healthy disc, it's lower than other tissues, and then as there's an issue with this nutrients flowing in and waste product flowing out, it starts to decrease. So it's linked, the pH is linked through that cartilage end plate to the blood supply. And then as the disc becomes more degenerate, it could actually start to rupture, or you may get disc herniation, at which point the contents could be spilled out into the body as well as it gets worse.
I just have a question. So this is human work.
Yeah.
Are you supposed to do some animal work before to try to human, or because this is cells and this is stem cells? So do you need to work on rodent people to access to human?
I think, so although this is all human, it's mainly done in vitro, so this is kind of outside of any type of model. So the next step would be, first we'll work on a bovine tail model where we have whole motion segments in culture. So we can put in human cells into these bovine discs. And there may be some issues between putting human cells into an animal model there. Other models which do exist in the field are things like rodents and rabbits. The problem is trying to get a model which simulates the disease process. So what they do at the moment is they may just like puncture a hole into the disc, and you get some of the same processes occurring, like inflammatory response happens, you get breakdown of some tissue, but you haven't got the issues of basically the nutrients transport. So it's really hard to try and mimic that. But yeah, this will all have to go into much more models and animal models before it could get anywhere near humans, which is good to know. We'll save.